So it's very important in the general public to make science, because this is increase awareness about what we do in the lab, first of all, and increase sensibility to fund research. And then, of course, people understand much more what is science about, so that we are trying, of course, to find solutions, but not only. I do believe that to bring science in the general public is a mission of a scientist, so we do not have only to stay in the lab, but also to make this available to everybody. This, in the long run, will push politicians to put more funding, and not only politicians, but also company and any foundation that can fund science. Differentiation is something that goes from one cell to an adult. So one cell can divide and give rise to two cells. This one cell is very totipotent. Divide and give rise to 2 cells, then 8 cells, and to the morula and the blastocyst. From the blastocyst, that is 3.5 days, and arrives to an embryo which is formed from 3 different tissues, which are called mesoderm, endoderm, and ectoderm. This little mass of cell, which is called inner cell mass, and this group of cells basically give rise to embryonic stem cells. We in the lab can isolate embryonic stem cell from this group of cells that derive from an embryo 3.5 days post-fertilization in a mouse or 5 days in a human. So these cells are very totipotent, so they can generate each tissue of the body. They can be propagating indefinitely, and they can differentiate in each cell of our body. Differentiation is when you take a stem cells, and this stem cell can give rise to erythrocyte, lymphocyte, muscle cell, or neurons, any cells. But this process that seems very easy is very tightly regulated. Normally what you have is that from a stem cell you have another stem cell that is called a progenitor cell or adult stem cell. So for example, an adult stem cell of the nervous tissue can give rise to neuron, oligodendrocyte, or astrocytes. For example, the lineage development of the hematopoietic system start from an adult stem cell that is called hematopoietic stem cell. And this, as you can see in these slides, can give rise to a plethora of different progenitor cells. These cells here can differentiate in these cells that is called CMP, and CMP can differentiate in MEP or GMP and so on. But the interesting things that I want to point you out is that at each branch, in the end, can give rise to this cell line or this cell line, so macrophage or eosinophil, but it cannot give rise, for example, to erythrocyte. There are adult stem cells that are multipotent, that is different to be totipotent, because they can differentiate in some cells and not in others. You can have adult stem cell in the brain, in the heart, in the bone marrow that can give rise to specific cells of that particular tissue. This is what is differentiation. It is a step-by-step process that a cell that is a stem cell become more and more committed until it becomes a differentiated cell. These cells, they beat as the heart. A stem cell that we have induced to differentiate in a cardiomyocyte in the lab. Differentiation is something unidirectionally. From a stem cell, you can go to a muscle cell, to a fibroblast, to a cardiomyocyte. You can go to a fibroblast, that is a skin cell, and then you can go back to a stem cell, and then you can differentiate again this cell in another lineage. So this was really a change of the paradigm. I mean, up to last century, it was thought that differentiation was unidirectionally, but this is wrong. So reprogramming is when you go back from a somatic cell to a stem cell. It's an increase in potency or the differentiation. And therefore, I mean, we know very little about we don't know if it occurs in nature, and at the moment, as I said, is a technical laboratory to produce, let's say, stem cell from somatic cell. One of the very early experiments demonstrate that the fate of a somatic cell can be reversed back to a stem cell phenotype. And this was through nuclear transfer. You can transfer the nucleus in a nucleated oocyte. What happens is that you generate a clone, so the nucleus of the differentiated cells is completely back to a stem cell phenotype, but can also generate several clone animals. So in this way, we have changed the fate of the nucleus of the somatic cells and made possible that this new entity, the clone, can generate a new organism. There are other methods. One is that you can basically make an extract of stem cell or make an extract of oocytes, and then this you can basically put on somatic cell, and this you can induce partially a reprogramming, not stable, but nevertheless you can induce some reprogramming also by this method. Another method, that is through cell fusion, you can take a differentiated cells and you can really fuse with a stem cell, so then you have an hybrid here. There was an experiment in a lab from Dr. Schuller. They took the cytoplasm without the nucleus, and this was fused with a somatic cell, and in this case they did not get any reprogramming. In contrast, when they fused the nucleus of the stem cell with the differentiated cells, they got reprogramming. The factors that induce reprogram are in the nucleus of the stem cells and not in the cytoplasm of the stem cell. You take this gene and you put this gene into the nucleus of the somatic cell. What you will have is that with very few genes you can convert this cell into a stem cell-like, or what is called iPS. This was the experiment of Yamanaka. They took a differentiated cell, they expressed these 4 factors here through a virus, and then they could generate an iPS cells that give rise to, could be propagated like embryonic stem cells, or they could generate mice. There is another method, which is the lineage reprogramming. You can make a direct conversion from one cell to another without passing through the pluripotent state. So this method have been identified specific factors which can do this conversion. To conclude this part, to wrap up what I said, it was thought for a long time that these are all, separated box. From an embryonic stem cell, you can have ectoderm, mesoderm, and endoderm, what I told you really at the beginning, and it was thought that there was not possibly, way of passing from one box to another. But more recently, with 2 different approaches. One is through cell fusion or through the expression of specific factors. Now we know that you can push differentiation back and you can really pass from one differentiated cell into another. Now, what we can do with this? There are several applications or dream projects, and one of the dream projects is that you can create iPS and you might cure a sick organ. So this is a dream project because it's really, we are very far to reach to this point. We are studying the mechanism controlling reprogramming, and so there are a group of genes that in the balance between differentiation and stemness, they maintain the stem phenotype. There are a group of genes that maintain the differentiated phenotype. What we try to do, we try to silence the genes that induce differentiation to keep the stemness phenotype. These are the 4 factors that the Japanese researcher Yamanaka used in his first experiment. These are not essential, so they can be substituted by other factor or by other modulating other pathways, so they are all not essential. At the moment, the use of this IPS is in the clinic is not even in the clinical trial phase. You can find also these 10 top things to know about the stem cell treatment. There are different types of stem cells. There is not a universal stem cell therapy that will cure any types of disease or any type of conditions. There are very few accepted stem cell therapy currently. Just because people say that stem cell help, this does not mean that they do. Why it takes so long to develop new therapy? Because science is going very slow. When you do a stem cell therapy, you have to instruct the cells to become what you want they have to become. As much as we manipulate in vitro, in culture, as much as we induce differentiation, and therefore cells lose their potency, and they likely is more complicated to handle and to let the cells to do what you wish they should do. There are a lot of treatment for sale that are not are very, very risky, also because clinical trials take a lot of steps to authorization to go really into the clinic. Nevertheless, we do believe that stem cell science is an important field, and we hope that in the end we will really have something important for regenerative medicine. We are mostly concentrated in cell fusion mediated reprogramming. Why? Because it's a very, very efficient way to induce reprogramming. We do believe that it might be a physiological mechanism. Cell fusion is a process that occurs normally during development. It occurs during fertilization. It occurs during the formation of a specific tissue in the placenta, or during the formation of muscle, of bone. Some people do believe that fusion can be a mechanism to repair or regenerate a tissue, and we are studying exactly this. So what is regeneration, first of all? It's something that in the lower eukaryote, like in fish or in amphibians, it happens naturally. And one example of this is a particular frog, is the salamander. After the amputation, these cells here, they undergo dedifferentiation, and then they redifferentiate again, and in the end they form again the limb. Of course, we don't have this ability to regenerate our hands if we have an amputation, unfortunately, but this means that during the evolution, this is something that have been lost from lower eukaryotes to higher eukaryotes, but can in a way be stimulated. And so we are trying exactly this, to stimulate this process. What we are doing in the lab, and is our working hypothesis, is that if you have a damage somewhere in your body, you can have some circulating bone marrow cells that can fuse with cells that have been damaged. To make it easy, it is a signal that can activate the cells and tell the cells, look, you have to become very potent and you have to activate this group of genes, okay? We do believe that there is a reprogramming of the hybrids that are generated after cell fusion, and then finally a regeneration of the tissue. We have done this in vitro, and now we are using a lot this approach to try to regenerate or to cure or to enhance regeneration in several disease models. What we believe is this schematized here. Researchers that thinks that can differentiate or become something different in other tissue, or after fusion can become something different. But we do believe that a cell from the bone marrow fuse with a somatic cell and form this hybrid, which then can be reprogrammed and regenerate tissue. To conclude, Sandro Botticelli is a painter of the Renaissance in Italy. We do believe that reprogramming is like the Zephyrus that blows on the cells, and these cells, they can be regenerated as the Venus here represented, and we do believe that this painting represents a bit the process of reprogramming.